Mutations are important to talk about. A mutation is an error in the base sequence of a gene that's reproduced during DNA replication. So you have a mistake. Now, mistakes are very rare, but they do happen. So the mistake happens, and you end up with a DNA with an inaccurate or incorrect base sequence in it. Then that DNA replicates itself and perpetuates that incorrect sequence. So this can cause changes in amino acid sequences during protein synthesis. So if you get a base sequence change in the DNA, it can cause a problem in the protein that's made from that part of the DNA. Some of the mutations that occur are, what do they call them? I want to say benign, but I don't think that's the word. There are mutations that don't end up causing any problems because it just doesn't end up mattering. Because those codons for the proteins, the amino acids, most of them have several different codons. And so if your codon was, let's just use an example. Like if you had ACC and ACU, I don't know this, don't quote me on this, but let's say that those two coded for the same protein, same amino acid. If you ended up with, if you had a mutation where this C was changed for a U, it wouldn't end up affecting the protein at all, right? Because it's still coding for the same protein, even though there's a mutation there. So sometimes mutations have very serious consequences. They can cause death. Sometimes they cause mild consequences, or sometimes no consequence at all. When we talk about a mutagen is a substance or an agent that causes a change in the structure of a gene. And examples of these are radiation and various chemical agents. So radiation, like from X-rays or other high energy radiation, can cause mutations in your DNA. And that can cause problems, can cause things like cancer. Chemical agents can also do this. Here's an example of how cytosine could be changed into uracil. So here, this is nitrous acid, and nitrous acid can react with cytosine to convert it into uracil. And I don't know if I think I must have done this subconsciously. Look at what I did over there. That's kind of scary, actually. Here I said, well, what if a C changed into a U? That's exactly how it could do that. I didn't even look at that. So I'm just not even going to try to take credit for that. Your book, which is really, really thorough on lots of other things, is really sketchy on the mutations thing. There are different kinds of mutations, and I'll try to explain this to you. First I have to actually open the drawing window. Well, let's stick with the DNA. So let's say we had this sequence. A point mutation is where one gets changed. And so if this was the parent strand, then on the daughter strand we might have T, T, and instead of C, maybe we got A in there somehow. And this one would be A, and this is C and A. There's a mistake here, because A is not supposed to go for C. Now that can arise in many different ways, and the whole replication process has a lot of checks in it that prevent this from happening, but it still happens occasionally. So a point mutation is where you've got one base that's incorrect. That's a point mutation. Point mutations are less likely to be a big problem. The other kind of mutation that you can have, which I can't remember the name of right now, that's really sad. And it won't be on the exam, but I just want to tell you about it. Another problem you can have is where you lose an entire base. So maybe that one got lost. And so then what you end up with is TTACA. Well, when you're looking at codons of 3 that code for an amino acid, if you've got this gap here, that means everything after it is wrong. Something shift, that's what it's called. Base shift, perhaps. I can't remember, I should remember that. But you can have that, or you can have something get inserted, and those have much more serious consequences. Your body also has repair enzymes that, like if pairs get incorrectly paired, the enzymes will come along and they will fix them and actually repair the problem. So let's predict the effect of a point mutation. So if CTA is point mutated to CTT, what's going to happen? Well, it depends on what those are codons for, right? I don't think we need to actually get the genetic code out and look at all of these. But this is talking about the DNA base segments, and so the RNA would be the opposite, right? And this is where we ran into some trouble last week. So T would be with A and A would be with U. And then that's backwards, so at least I think it's backwards. So this is the codon. And then this guy, C, is with G, T, A, and T is A. And so the codon that would arise from that one is AAG. So the effect, or the changes in the amino acid identity, are going to depend on whether these code for the same amino acid or for different amino acids. If they happen to code for the same acid, there's going to be no change. And that's entirely possible because there's a lot of duplication in the genetic code, which is a good thing because it makes these point mutations less of a problem than if every codon specified, if every amino acid only had one codon. Does that make sense?